We can use biotechnology to create genomic libraries. So just like a library is going to be a collection of books, our DNA libraries are going to represent a collection of cloned DNA samples. And you can take these from pretty much anything that you'd like to study. So as long as they're coming from a single source. So you could look at samples from a particular tissue type, like maybe liver cell, or liver tissue, kidney tissue, heart tissue, lung tissue, so on. Cell type. Okay, so different types of cells. You could look at different type of blood cells, or you could look at different types of liver cells, so on. Or samples from a single individual that may be coming from all different types of cells. A genomic library is going to contain at least one copy of all the sequences in the genome of interest. Genomic libraries are constructed by cutting that genomic DNA with a restriction enzyme. All of the pieces are then ligated into different types of vectors, and those are chosen depending on the size of the genome. So if you're trying to look at something with human cells, you might use BACs or YACs because they're going to be much larger and able to take up larger pieces. If you're looking at something like a bacterial genome, you might be able to get away with cloning those into various different types of plasmids. These YACs indeed were commonly used to accommodate large sizes of DNA, necessary to span the 3 billion bases of DNA in the human genome. There are different techniques, though. We'll talk more about those. Things like the whole genome shotgun cloning approaches and different types of sequencing methodologies like next generation sequencing. And these are replacing traditional genomic libraries. So rather than sequencing the human genome piece by piece, you can sequence the whole genome at one time. In these, though, the genomic libraries, all these DNA fragments are sequenced, and when you're doing these whole genome approaches, you don't need to clone them into these host cells. cDNA libraries, or complementary DNA libraries, are going to contain DNA copies that are made from the mRNA present in a cell. So what it represents are all of the genes that are active at the time the cells were collected for mRNA isolation. So what you can do is you can collect cells, you can treat them with different conditions, and then perform this technique so you can see what were the RNAs that were active under this condition, or what were the RNAs that were active under that condition, and compare them. So the way that it's made, again, you have your mRNAs that you were able to isolate. All of these RNAs are going to have the poly A tail and the 3' end. You add an oligo DT primer. This is just a series of T's. They're going to attach to that poly A tail. There's an enzyme called reverse transcriptase. What this will do is it's going to make a DNA copy of your RNA. This is an enzyme that was isolated from viruses such as HIV that have RNA as their primary genetic material. They perform a similar function. They need to go through this DNA intermediate in order for them to replicate. So we use the enzyme for this process. We take advantage of the enzyme for this process to make this DNA copy of RNA. It's kind of backwards from how we normally think, though. So the DNA synthesis is going to continue. You can partially digest this RNA with this RNase that will digest it. Add DNA polymerase. You need to leave some there so that the DNA polymerase has something to start off of, to start from. It'll fill in these regions here. There's still going to be a few little gaps, remember, and you need to add DNA ligase to then seal those gaps. At the end of this, you're going to have regions of double-stranded cDNA. So this is going to be DNA, again, complementary to those mRNA present in a cell. And then the same process happens for all of the mRNAs within a cell at the same time. PCR is a technique, or polymerase chain reaction, and it will copy a specific DNA sequence through in vitro reactions. And what it does is it amplifies your target DNA sequence, and these are usually going to be DNA that's present in very small quantities. So it's a nice technique. It's a very rapid method of DNA cloning, and it's going to eliminate the need to use host cells for cloning, or is some of the intermediates at least. So it starts out, cycle 1, you've got your DNA that you want to amplify. You denature it. The way that you do this is you heat up the DNA to about 95 degrees Celsius or so. This will cause the hydrogen bonds between these two strands of DNA to break. you then have primers within this solution. Primers are going to be short regions of single-stranded DNA that will bind to the forward and end of your target sequence. You drop the temperature, so this will allow these primers to anneal. You can then add a polymerase. There's a polymerase called Taq polymerase, and this will work at even very high temperatures. So what you do is you raise the temperature up just a little bit. This Taq polymerase is in here, and it will extend these sequences. So at the end of this cycle, you will now have 2 pieces of DNA that's going to be the same as the region between the 2 primers that you have selected. After cycle 2, the same thing is going to repeat, and you're going to have 4 copies. And you usually repeat this for about 30 to 35 cycles. And after about 30 to 35 cycles, you're going to increase your number of DNA copies by quite a lot, maybe up to 1 billion. So some of the benefits of using PCR for different techniques, you can use allele-specific probes for genetic testing. So if you want to see if somebody is a carrier for a particular trait, you can use a small sample of their DNA. You can run this PCR technique. It takes about maybe 3 hours to run, and it's a pretty quick way then to help to diagnose these genetic disorders. It's also very advantageous when you're studying samples that are in very low quantity. Maybe you only have a single cell. Maybe there's a fossil with very little remains. Maybe there's a crime scene where you have found just a single hair or a saliva-moistened postage stamp. Okay, these all can potentially be sources of DNA for this PCR technique. You need very, very little to start with. About maybe 1 microgram at most of this DNA to start with. So a con of this is that you must know at least something about the nucleotide sequence of your target DNA in order to design primers that will bind to that DNA. Those primers need to bind at pretty high fidelity, and the primers are usually about 20 to 22 nucleotides long, so you need to know at least that much about the sequence. If you have even minor contamination from other sources, that can potentially cause problems as well. It also cannot amplify long segments of DNA. So PCR starts to fail if you try to do longer than, maybe about 1,000 kb, 2 kb, something like that. You can't go too long with the segments that you're trying to amplify. RT-PCR and qPCR are 2 other techniques that are used to look at gene expression in a cell. Reverse transcription, or RT-PCR, is used to study gene expression by studying the mRNA production by cells or tissues. So you've got your DNA in here. It's going to go through the reverse transcription process to make, so the cDNA is made. After that, there's this quencher and probe that are on here. When the primer comes through, when the polymerase comes through, it's going to light this up. As it continues, it's going to eventually hit this quencher, and that's going to make this level go down. So when you're doing just simply the RT-PCR, this is going to tell you whether or not a particular mRNA is present or not present in your cell. If you're doing quantitative real-time PCR, or qRT-PCR, this will allow you to determine the relative levels of that mRNA. So it's a little bit more quantitative of a technique. Rather than a yes-no answer, it'll give you more of a level. So if you look here, you can see something that's a very high gene expression, and you can compare it to something that's very low gene expression. So a way that I've used it in cells is I've treated cells with different types of drugs. I harvest the mRNA from those cells, and then I look at the expression of a particular gene. Is the level of that gene high after that treatment with those drugs? Is that level low? And how are those levels relative to one another?